Plastids, definition, classification, structure, functions by Dr. Kushnud or Raymond. Plastids are a double-membrane diverse group of physiologically and genetically related eukaryotic organelles that play important roles in plant metabolism through different processes like photosynthesis, nitrogen and sulfur assimilation, amino acid synthesis. These plastids are found in plant cells and algae. Hence, they differentiate plants and animal cells. The term plastid was given by Schimper in 1885. Plastid contains a double-stranded DNA molecule that is circular and resembles the circular chromosome of cells of prokaryotes. Classification. Proplastids, undifferentiated plastids, may undergo differentiation into many forms, depending upon their specialized functions in the cell. They may develop as following variants: Chloroplasts, chromoplasts, leucoplasts, gerontoplasts. Chloroplasts. Chloroplasts, green plastids, are 5 to 10 micrometers long organelles. They are surrounded by a double membrane known as the chloroplast envelope. In addition to the double membrane, chloroplasts have a third internal membrane system, known as the Follicoid membrane. They are located in the mesophyll of plant cells. They perform the process of photosynthesis and synthesize their own food. Other functions carried out by plant cells include synthesis of fatty acid, synthesis of amino acids, plays important role in regulating the immune response in plants. Structure of green plastids, the chloroplasts. The majority of chloroplasts of green plants exist in the leaf mesophyll cells. Their shape resembles discoid, oval, spherical, or ribbon-like organelles. Their length varies from 5 to 10 micrometers and their width varies from 2 to 4 micrometers. These organelles are encircled by double membrane. The inner membrane of chloroplast is comparatively less permeable. Stroma is the space surrounded by the internal membrane of the chloroplast. This internal space is filled with fluid that surrounds the grana, follicoids. It also includes ribosomes, starch, and DNA. A number of flattened membranous sacs are organized and are called the follicoid. These are located in the stroma. Light-dependent reactions of photosynthesis occur in these follicoids. There are packets of chlorophyll pigments in the follicoid membrane. These are termed quantisomes. There are 230 to 250 chlorophyll molecules present in every single quantisome. Space surrounded by the membrane of the follicoids is called a lumen. This lumen is the site for the phosphorylation process of photosynthesis. During light reactions of photosynthesis, the pH of the lumen becomes acidic due to the inward pumping of protons. Follicoids are arranged in a bunch that resembles the heaps of coins known as grana. A single chloroplast contains 10 to 100 grana enclosed in it. Along with these grana, there are membranous flattened tubules called the stroma lamella. The stroma lamella interlink follicoids of grana. Stroma. Chloroplast contains essential enzymes. These enzymes play a key role in the synthesis of carbohydrates and proteins. Stoma also contains other structures like ribosomes and small, circular DNA. Follicoids also contain chlorophyll pigments. The ribosomes present in the chloroplasts are smaller, 70s type, than the ribosomes of cytoplasm, 80s. Etioplasts. These are precursors of chloroplasts. These arise from proplastids that are not exposed to sunlight. Upon exposure to sunlight, these etioplasts are converted into chloroplasts. They usually exist in leaf tissue and stem angiosperms that grow in extremely low light conditions and darkness. Chromoplasts. These are colored plastids. Their main function includes pigment synthesis and storage. They exist in flowering plants, fruit roots, carrots and sweet potato, aging leaves i.e. senescence, and are responsible for their color. Their distinctive color formation is due to carotenoids. Chromoplasts synthesize and store pigments such as xanthophylls, yellow, carotene, orange, and many other red pigments. Through these distinctive colors, they attract pollinators to the plants. Leucoplasts. These are unpigmented i.e. colorless plastids. They are found in non-photosynthetic roots, bulbs, tubers, and seeds. Amyloplasts. These are double-enveloped organelles. For the plant, they store and synthesize starch through glucose polymerization. Leucoplast exists in 4 types. Amyloplasts. These are double-enveloped organelles. For the plant, they store and synthesize starch through glucose polymerization. Proteinoplasts. They are known as proteoplasts or aleuroplasts. They store proteins and exist in seeds, pulses. Eleoplasts. Eleoplasts is also known as oleosomes. They store fats and oils that are needed for the plant. They exist in seeds. Tannosomes. They are found in vascular plants. They synthesize polyphenols and tannins. Gerontoplasts. They arise from a chloroplast during the aging of plant foliage. Structural changes in the thylakoid membrane cause the formation of gerontoplasts. Inheritance of plastids. Plastid inheritance in many green algae and land plants is from a single parent. Generally, plastids are inherited from the maternal parent in oogamous species. Even if plastid inheritance is from both parents, it is generally predominated by maternal plastids. In conifers, plastid inheritance is from the paternal side. Even though there is strong maternal influence, there exists some species in which plastid inheritance shifts from completely maternal to absolutely paternal nature. Thank you. Like, share and subscribe.